This lecture is on citric acid cycle. It's also known as Krebs cycle based on the person who discovered it, that is Hans Krebs. It is also abbreviated as TCA, tricarboxylic acid cycle. But whatever it is commonly referred as Krebs cycle. So if there is any exam question, I will also use Krebs cycle as the means to ask question. So this is the citric acid cycle which we will go through, which has connection directly from glycolytic pathway, and the location wise citric acid cycle is to be happening in the mitochondria. To be specific, the matrix of the mitochondria. Okay. Here we have the citric cycle overview, but this is interesting because I plan to give you an understanding about if there is a question about pathway and how much you can actually answer. We are all bogged down in terms of ability to memorize. So in terms of this, this is a good lecture to actually give you an idea in terms of what you can give answer if a certain pathway has been asked in exam paper. For instance, in this overview, it is written in a very simple manner, where you only have a few steps being shown here, and then you will see in the next slides and the slides following, it will have more complex pathway being shown for citric acid cycle or Krebs cycle. Here, basically, to give you an overview, that it starts with acetyl-CoA, and it reacts with oxaloacetate, and 4 carbon react with 2 carbon, it will become 6 carbon citrate. And then you have decarboxylation. Every time there's a decarboxylation, you lose 1 carbon, therefore you get alpha ketoglutarate from 6 carbon citrate to 5 carbon, and another decarboxylation which will then yield succinyl-CoA, become 4 carbon intermediate. And then through a number of steps, it will eventually become oxaloacetate, and this is a cyclic pathway. As such, this goes on again and again. So just now, a slide of a simple Krebs cycle, which now been shown to be much more complex. So where you get to see all the 8 steps within the cycle itself, and also additionally one more step outside of the cyclic pathway. It is very much difficult to put this one step, that is pyruvate to acetyl coenzyme A. It's either to be in glycolytic pathway or that of the Krebs cycle, and eventually it is been decided to be combined with citric acid cycle. But do remember that once the glucose undergoes glycolytic pathway, pyruvate will be the end product, and pyruvate then enters mitochondria into the matrix, and then it will be converted to acetyl coenzyme A, and only then the citric acid cycle will begin, and it is a total of 8 steps. In terms of the 8 steps, just now I said it's very difficult in terms of remembering. In one way to do it is by using abbreviation. I normally remember or memorize it by using this abbreviation referred as OCIKSSFM, where O stands for oxaloacetate, where it reacts with acetyl coenzyme A, and then C is citrate, and then isocitrate I, ketoglutarate K, SS involves succinyl-CoA and succinate, and then FM fumarate, malate, and then back to oxaloacetate. So that's one way you can also memorize the steps relevant to Krebs cycle. In terms of Krebs cycle, there are three locations that will yield NADH from the reduction of the coenzyme NAD, and for each of the acetyl coenzyme A reacting to oxaloacetate and then undergoing the complete cycle itself. The location wise is at location 3, location 4, as well as location 8 has been put on the slide there. In terms of yielding FADH2, also at location number 6, you also have one GTP. The thing about GTP is it's only found for animal cells, but those of plant and microbe, it should be directly yielding ATP rather than GTP. But you need to understand each GTP is equivalent to ATP, one ATP without loss of any energy. So that's in terms of the number of NADH, FADH2, as well as one of the GTP. Other than that, you can see that there is a loss of carbon dioxide at two locations. Therefore, the number of carbon for the intermediate reduces at this location itself.